Thanks everyone for the invitation. Thanks a lot, Malar, and I guess Gabe, you've done as well. So this is not my show, this is their show. And I'm super glad, as well as Michelle, who's here. I just want to have one quick word of introduction. So I think most of you know already Seb and Nikhil, probably from like your past experience and work here. Michelle is a PhD student in my lab and is working a lot on the Nipoppy project that you will hear about. What I want to say is a lot of what is happening is coming from a sense of a need for a more like a community and standardization aspect of our work. Everyone seems to be reinventing and redoing and like in a way grabbing the tools and like and then PhD leaves and things are difficult to like capture again and do again and reproduce and replicate and all those things. So I think a lot of that is coming from this aspect. Another aspect of it is that if we can make standards and if we make some things that are common to the community, we'll be going slower, but we'll be going much farther. And at some other people will be going much faster as well. So I think that's another aspect of that work that is important to me. And I don't think I want to, nothing more than that. It's just like a lot of good things are already happening with others in the community, and I hope that this is going to be of interest to you all.
Thank you, JB. Thank you, Mallar, as well, for hosting us and Seb for planning this. The way this is going to work, I guess, is we're going to give a brief introduction of the things that we've been working on in our lab. So for like 20-25 minutes. And rather than boring you with more details on what we do, we're going to have a panel discussion to have different types of perspective on the challenges that we usually face when we are dealing with multicentric studies. We want to do fair data and reproducibility and open science type of things. It's a complicated problem with technical issues as well as cultural issues. So hopefully we'll get a broader perspective when we talk in panel. But more specifically, what we're going to talk about today first is a couple of things that we have faced over the last few years when it comes to data curation, processing, harmonization, sharing. And if you have all worked with data in any capacity, small scale or large scale, there are challenges like just naming of the data. That's as simple as it sounds, but people like to name their data as celebrities like to name their kids. So it's very messy to deal with. The processing, as you have known, if you try to reproduce somebody else's pipeline or workflow, it's very difficult. From the paper, you can never actually get all the details. And then harmonization and sharing, which comes more into play when you are trying to compare data from one study to the another study, and simple things like how do you encode values in your Excel sheet or CSV, just how do you encode diagnosis, those type of things become very critical. And for those things, you definitely need a clinician in the loop for things like how do you calculate different types of scores, assessments, that has to have a human in the loop. So it's not just technical, but also a human issue. So yeah, basically all of this takes a lot of time, and that's the gist of this. It's inevitability of manual work that we have faced over the last few years, despite all the progress in technology. So what we really want to do in our lab is come up with solutions that are easy to use, and start with the one single data set or one single study or one single lab is the unit where we can deploy this type of tools, best practices, protocols, frameworks, whatever you want to call it. But at the end of the day, something that makes your job easier to do your research. And how do we scale that up to multicentric? So that's where going from one study to multiple studies, how do we coordinate things together? So the first thing we have is basically Nipoppy, which basically deals with single study or single data set level, whereas Neurobagel will more talk about multicentric or multi-data set problems and solutions. So as this diagram shows, basically Nipoppy is a series of steps that we implement at a local level, and then Neurobagel takes that and deploys that into multiple studies or multiple sites, and we all kind of have a happy harmonized data sets to play with as we go along. I'm going to go through this bit quickly for the sake of time, but if you have any questions about the specifics, do let us know. We're happy to talk about this in later. The philosophy here is we start with the raw data. So it could be DICOMs for imaging or CSVs or Excel spreadsheets or REDCap instances, whatever you have, we start from there. And we want to help you think ahead, foresee the problems you may have if you don't take care of some data-related issues from the get-go, and that will make your life much harder for avoiding those in current state. So first thing we do is basically curation. So we extend bits. If some of you don't know what bits is, it's a standard for organizing data. And we extend that to cover not just imaging, but also clinical data, so tabular data more specifically. So everybody has the same type of organization on their desks locally. And that really helps when you have same organization, you can really share that much easily, query that easily as well. Once the data is organized, we also implement containerized pipelines. So what containerized pipelines, if you're not familiar, it's a wrapper that ensures whatever you're running on one machine will also run on some other machine, regardless of the setup you have of operating systems or Python or whatever packages you're using. So it basically packages things nicely. So if you run one analysis in your lab, you can easily share that with somebody else in other labs, or just a new student that comes to your lab in future can run the same thing without any issues. And finally, we also, I don't know how many of you have written grants recently. I'm sure JB and Mallar can relate to this more. We want to know how many subjects we have. And it is deceptively difficult thing to know because there are missing data, there are missing values for certain modalities, there are missing data types that you need to know beforehand. So just knowing what data you have, what has been done with that data in terms of the processing of that data, or which values are missing for clinical stuff like diagnosis, scores, sample demographic information. Because if you don't have demographic information, you probably won't be able to do most of the analysis either. So all of that needs to be somehow nicely tracked and gets very messy if you don't start from the beginning. So we have tools to basically keep track of what data you have and how it's been processed and what's available for analysis at the moment. So again, going quickly through, this is not some fancy tool. It's essentially some guidelines you follow as a student or as a ECR or a PI in the lab, and which simply creates some framework simply creates a directory tree on your disk. So we start with like an empty data set. So think of you want to work on a new data set. You start with some cookie cutter layout on your disk, and you start populating that layout. And the tools we have created in Nipoppy basically let's help you do this automatically. So you don't have to keep track of that in your head, and everything gets created automatically and populated automatically as long as you tell it what data you want to process and what data you want to curate. Again, I'm not going to go into details for the sake of time, but the idea being we give you tools to organize your data, imaging as well as clinical. We give you tools to basically specify what type of pipelines processing you want to do on your data, and we keep track of that using this global config file. And then we basically run some certain pipelines to organize your data directly into bits, and then run pipelines to basically give you derived data. So if you're running MaggetBrain, for example, you want to have some volumetric features from your MRI, and you want to know how many subjects were finished successfully and how many failed or why they failed, we can help you track that. So you only rerun the things that didn't go well. So that's kind of the gist of the Nipoppy. It's a protocol in a way and a framework that guides you organize and process your data. And also tells you what has been done to your data, so you can be sure that what you have is real on the disk and not just in your head. You can also share that with other people in your lab, so they don't rerun the same things. And also, basically, in the papers you write, you're 100% sure that you can rerun the same thing again, and it will give you the same results. Because there's nothing scary than the reviewer comes back and tells you to rerun something and you don't know what you did 6 months ago. This is a small dashboard demo that we have. So once you start from the raw data and you do all of this Nipoppy stuff, you basically get this nice interactive dashboard on the web, which is running right now. Anybody can use it. And it will basically give you an overview of what data participants you have, what modalities you have, whatever processing you have done, and how many things completed successfully, didn't finish successfully, all that stuff in like one web dashboard. So when you want to know what has been done and what needs to be done in future, you can easily do this interactively in a dashboard. So just to take away points for Nipoppy, which again, it builds on existing tools. So if you're familiar with bits, we are not reinventing bits. We are just extending it. If you're familiar with AppTenor or Singularity or Docker, which are the containerization technologies for running your analysis, so we are using that. And Bootix is another tool that was developed by Tristan Gattard in Concordia, which tells us what parameters were used or while you're running your pipeline. Sometimes you get failures and you don't remember what commands you ran. So you could do either Control R for bash or look for your notes in some file to see which exact command you run. And that's probably not a good idea as you go forward. So all of these tools help you make your research more reproducible and easily shareable. Yeah, so I just said it helps with the organization and reproducible processing of the data. We also have tools that help you, like I said, track what's been done, also give you individual level features that will actually go in your statistical model because you don't really deal with actual MRI all the time. So IDPs are image-derived phenotypes. So you get this nice tabular file saying, these are the features I want to use in my analysis at the end. And the most important thing, this is not fancy. This is really simple. It's a decentralized thing on your disk. There is no fancy downloads you need. And at the end of the day, what it gives you a nice small CSV or digest or bagel, as you'll see more about this. The bagel that you can share around with other people. And it will tell other people what data you use, what processing you did, and what features are you using for your analysis, and the one CSV. So if you want to reproduce your data results, or if you want to reproduce somebody else's results, it's a nice single shareable bagel for help you. And I'm going to stop here. But in the future, we basically want this to be a more community thing. We want if you all do the same thing, it's better for all of us. And now I'll give it to Seb to tell you how this scales into multiple studies.
Thanks, Nikhil. Yeah, so just taking a step back, zoom out, so to say, what Nipoppy does is it gives you confidence about one data set, and those stages that Nikhil was showing here are kind of modular. So you can come in at any point, but it gets harder the later you come in. If you start from the beginning, your data will look good at the next step already. And so what you can do is you can do the kinds of things like Nikhil said. I want to write a grant. How many PD Parkinson participants with these demographics, these imaging features, those IDPs do we have in this data set right now? That you can answer. But often we have more than one data set. Ideally we do. And in that case, it's not enough to do the internal harmonization or internal curation of one data set. Now you need to combine them. You need to harmonize across. And that's where we switch from Nipoppy to Neurobagel. So now it's purple, you will notice. And the thing that we do in Neurobagel is kind of the same philosophy that Nipoppy uses, which is don't get in your way, don't change the way you already do work, just give you a little bit of helping tools to lower the technical barrier of entry to adopting these best practices and make your life easier somehow. So it's not just aspirational, it's supposed to be helpful. So in practical terms, your data stays where they are and the harmonization just gets added next to it. And I'll show you in a second how we do this. But if for now you just trust me that that's what happens, what this allows us to do is that we take your raw data and we have a translation of your individual data set into a common representation of the data. So let's say we all agree to describe diagnosis with ICD-11 codes, but each individual data set does something else, right? So there's your unique data set to ICD-11 mapping that happens through an annotation process. Now I'm going to show you this in a second. So I can now ask the same question I asked before, but across two data sets or many data sets in my institute. And the nice thing is, if I have a colleague in another institute who I cannot talk to directly, I can't see their data, but they're doing the same thing, I now have a machine that is over there that I know speaks the same language as my machine. So instead of sending emails around for answering this question, I can just make a single request and get the answer right away. So that's the big idea. And first question you probably have is, well, how do you harmonize it? Because that is hard. And that's where we pay a lot of attention. So the world where Neurobagel lives is the world of federated data. So that means data that cannot move out of, let's say, a hospital or a research center for data governance reasons, but you still want to collaborate with people. And in our model, the way we do this is everybody keeps their data locally. So we don't move them into a central location. We don't give them to somebody else necessarily, but we just add this annotation. And because this is a challenging thing, the way we approach it is through 3 steps. The first one is standards. So that goes back to the idea that Nikhil was referring to. We don't try to reinvent things. There's already an ICD-11, or in our case, there's SNOMED CT, which hospitals use for billing. Smart people built this for several decades. That's good. We can take that. That's a language for describing data. The next problem is you need to agree on what data you want to harmonize. That can be very challenging. So what we do is in Neurobagel, we say we're going to start from something very simple, something use case driven. We all agree on some very basic demographics, some imaging parameters, some cognitive assessment tools, and we start from there. But then the next problem is, okay, if you have the language and you have the variables you want to annotate, the annotation itself is technically challenging. Somebody needs to go and write JSON, XML, all kinds of different file formats. That is not easy, and especially not if you're talking machine understandable annotations. So we wrap this in a graphical user interface so you can give it to the people who really know the data, which are the clinicians, because you don't want your data engineer to explain what diagnosis this patient has, because she might or he might get it wrong. So the way this works on a high level is site A, site B both have a big Excel data set with a column, and in site A it's called DX, and in site B it's called group or something like that. I'm sure we all have seen this. And so we now ask procedurally a question, well, can you please point to the column that contains information about diagnosis? And everybody can do that if we make this easy. But that's not enough. The next question we have, because we want to have participant level information, we want to make cohorts across sites. So we can say, well, it says Pat in your column and it says 1 in the other column. Can you please explain what these numbers refer to? And this is the step where for the user, it's supposed to be easy, but something relatively big happens, because now we don't turn Pat into Parkinson and we don't turn 2 into Parkinson, we turn it into the ICD-11 code. So now there's no more ambiguity. We know both people mean the same thing. And so because we're doing this with data on the network or for federation, you can just swap out ICD-11 with SNOMED CT. That's the same idea. There's a controlled vocabulary for these things. So here is a short GIF video of how this works. It kind of started in the middle, so that's not the most useful. You can also go to this address and try it out yourself. But what we'll see in a second is this first step that I was talking about, where you say, can you please point out the columns that contain information about age, sex, diagnosis, and so on. So here I loaded a table, and what you see on the left are my variables that I can annotate. And what you see on the right, oh, you see something else than what I see, okay. Well, ignore what I said, but it starts by you painting variables to columns, right? That's the association. My variable diagnosis can be found in these columns. And then you go down to the level of values and you annotate. We can look at this more later. The big point here is it's graphical, it's relatively intuitive. You don't need to understand data engineering, graph databases, all that kind of stuff to do this. And that's the key, because you need to know the data. You are a scientist, not a data engineer necessarily. Okay, so the next question is, let's say everybody does this, how can we ask each other questions? And that's where I think our life gets a lot easier, because this is more of a social problem than a technical problem. Those technologies exist, have existed for a long time, but they are still hard to learn. And again, the people who want to use the data are not the ones who are supposed to learn that stuff. That doesn't make them any better at being researchers or clinicians. So here again, we decided that we need a graphical interface. So there's a tool that you can go to and essentially say, I want a cohort with these characteristics, and then you have access to all these variables. But what happens under the hood is this tool writes a query for you in a query language that is federation enabled, let's say. And then it sends it to all the places that are currently talking to each other, and the results come back. And then it shows you the results, right? So we're talking about clinical data. So the next question you must have at this point is, well, how can you do this if the data aren't public, right? Like how would somebody federate with you if the data aren't supposed to be open? And this is just a bigger presentation of this query tool. So that is a 2-part answer about how do we control access to the data. The first part is what we have right now, and the second part is what we're building. So what we have right now is when you zoom in to one of these bubbles, you get what we call a Neurobagel node. And that's something that's running at the in Molar's lab now. And what that means is just that there is a technical piece of software running that belongs to one person, and that can be a legal person, but the important thing is that's the data owner, and the data owner is responsible for data stewardship, data governance, access control. There can be many data sets that were annotated and belong to this node. At the moment, those are just data sets in Molar's lab, for example. But there's one person who has to, or should be able to decide who can see what's in this box, so to say. And that's the data owner. So at the moment, what this person can choose, and I think it would be fair to say that that's Gabe, maybe. What this person can choose is that it doesn't respond at all, so only Gabe can see it. It only tells you, yes, I have something that fits your criteria, or no, I don't have anything that fits your criteria. It can say, I have this many numbers that fit your criteria, or I can show you, hey, this is everything I have on the people that fit your criteria. And that is not enough in some scenarios, but that's what works right now. So if you scale back out, what this allows you to do is every site can choose this at any time and change it at any time because the data are still local. So if you decide tomorrow, my node is no longer going to talk to you, which is, well, that's no longer true of your node because you're talking to the MNI now, but let's say the NIMH node, for example, right, is not going to talk to anybody at the moment, but they can still search their own data, and then they can see everything that's in the public world, right? So I'll explain that in a second. But the idea here is every participating node can choose the level of visibility of data independently, and then we can talk about what that means for the end user, but they essentially just see less or nothing. And we also have fully open data. This is what you can search when you go to the open search interface, query.neurobagel.org. And I'm going to show you very briefly what this is. So in Montreal, we have a couple of groups that we work closely with. Obviously, Malar's group and the Douglas, which I think is the most running. But then the blue ones are the more clinical ones. So in the MNI, we have the Quebec Parkinson Network and the Quebec Parkinson Network from Alain Daguerre. Uri Monchi at UDM has the C-Open Network, also Parkinson Network, which is our initial use case. And then we zoom out a little bit more. Then in Canada, we have a collaboration with the Ontario Brain Institute, which is going to become very important in a second when I talk about the other answer to how do we control access. And what you mostly will see when you search on the open side is the OpenNeuro data from OpenNeuro. So essentially, OpenNeuro has a node in this model. And actually, if you go on the OpenNeuro website right now and you search there, you are not able to search for individual participants. You search for data sets, but they now have a little link that says if you want to do the individual search, you click here and you go to Neurobagel. And we can talk more about that. And then I think what's really interesting is that this kind of model allows people who cannot share data on OpenNeuro or cannot transfer the data to the MNI to talk to us. And one really important collaboration here that I want to point out is with the NIMHANS Institute in Bangalore, which Nikhil can say the most about because he has done all that work. And then Enigma is another important one, I think, where there is data that a consortium can really benefit from this, even if they don't talk to other people, right? Because within the PIs in a consortium, this is the same problem. You need to ask these questions of each other, and if that is an email or if that is somebody's time, that makes it harder. And now for the second part of the answer to the authentication. So what we really want, and that's something we've talked about a lot as well with Gabe, is we need a way for you to ask the question, and then the node that you're asking the question of should know who you are. So they should trust your identity. So there's essentially two pieces to this. There's I need to know that it's really Gabe who's asking. That is different from, I don't know who's asking, somebody. But maybe Gabe doesn't have access to all the data sets. So that's the second problem, which is I trust that this is Gabe, but I'm only authorized, or I only authorize Gabe to see these first two data sets. So these are two problems, and we address them separately. The authentication problem is tricky because we need something that works in a decentralized network. So we can't just give everybody an account because everybody, there might be a new node tomorrow and then they have to make new accounts. So the way we do this is through a federated authentication process. And there we rely a lot, and that's where the Ontario Brain Institute comes in, on, again, existing standards from the Global Alliance for Genomics and Health, who have thought a lot about this. And essentially the idea is you get a passport, right? So nobody needs to know who you are. They will trust your passport because they trust the government that gave it to you, right? So you can show up anywhere. They trust that that's really you, which is the authentication part. And the authorization part is a different thing. That's the visa. So some countries may not let you in, even if they trust that that's really who you are. And that's the authorization problem. And that is something that each individual node will have to deal with. Okay. This is the big overview. We have a nice website you can go to, lots of documentation. There is a group, and we're not even listing everybody on this group that works on this. We're working very closely with many collaborators. I think this is the right time now to switch over to the panel part of the discussion. So I want to introduce some of our panelists who maybe could join us on the table. And just for context, the reason we decided this was a good idea to present this type of work is because I think D3SM is a good example of a form of capacity building of different views on the same problem, right? And I think it would be nice to benefit from this by having all these different views at a table and discuss this problem. So I'm just going to do a quick introduction. So Matthew Denuric is a PhD student in Mallar's lab, also co-supervised with Yasser. And you are studying clinical and neuroanatomical profiles of patients expressing early psychotic symptoms. That is what you said. Okay, yeah. The next person I want to introduce is Professor Kate Levine, who is an assistant professor at Douglas McGill University, and her research focuses on cognitive dysfunction and psychiatric disorders, and also on developing open source digital tools to improve cognitive assessments. And I believe you also are leading the Douglas Open Science team. Next person I want to introduce, I'm going to skip you because, well, actually, no, let's do that. I'm going to introduce Genevieve Moore next, who is been working at the Douglas since 2011 and first built the Knowledge Translation Program and then the Open Science Initiative in collaboration with Dr. Isabelle Basselar, who's sitting in the audience, and more recently, Dr. Kate Levine, who is sitting at the table. Genevieve is now involved in activities surrounding data sharing and reuse, coordinating an alliance between 3 mental health research centers in Quebec, and you are also a CS employee, which I think is super interesting to have at this table. And the last person I want to introduce is Dr. Gabriel Deveni, who is the man of many hats at the Douglas, leading the DNP, which provides infrastructure consulting and software development for scientific computing, while also supporting the CIC and the Cobra Lab and all things neuroimaging, statistics, and on the side, leading his own neuroimaging research program. All right, so thank you very much, everybody, for joining us at the table. And the first question I'm going to ask to everybody, and maybe we can just go from left to right and just have a roughly up to 2-minute answer. Behind you is this pyramid that I like a lot about culture change. And I think we've been saying this over and over. This is less of a technical issue, more of a social cultural issue. What are the pain points surrounding fair and open research, whether you're doing the research or you're enabling the research, that bring you to the table other than the invitation? And do you think you can situate yourself somewhere in this pyramid, or maybe everywhere, right? So that would be my main question. If you could maybe just characterize where the things that we talked about impact you in your life, and yeah, Matt, you can start.
Yeah, for sure. So I guess I come from the perspective of working hands-on with data that like isn't always clean, and I think that's the experience of like everybody who works with data. It's kind of like the rule that it's messy, not the exception, right? So I think like, in my experience, if you don't have organized data or like a really good workflow for documenting what you did, you're like never confident in what you did, right? Like you always kind of have to circle back and say, you're looking over your shoulder, like, did I run this with the right subjects? You know, did I like exclude the right people here? Do I have to rerun this? Like you're always kind of going back and forth like that, right? And getting around that just takes time to build up that workflow and organize your data. So I feel like based on that, I'm at like the make it possible end of the pyramid. Yeah. But I do look at things like Nipoppy and stuff like that, where it's like literally built to get around that, right? It's literally built to make you more confident in what you're doing, where you can just like, without even trying to, have like a documented workflow for exactly what you did, and just have your data super organized. So Yeah, I'd say I'm at the possible end of the pyramid, but like the making it easy,
Yeah.
it's exciting to see tools like that can like really
Cool,
turn it
thanks.
around.
Yeah, maybe so. Katie, you're PI now, PI position. Where do you feel like this? You look
Yeah,
at this now.
I mean, make it easy is always the thing now for new PIs and even established PIs. I think the challenge is always time. So making it easy and making it rewarding as well for those that may not see the inherent value that some of us do in open science is important, but easy is kind of the first barrier to pass in order to make it available and something that looks exciting for people that might feel like, oh no, this is too difficult it's not for me. I wouldn't really be able to do it, but I want others to do it. I think making things like these tools and easy for others to kind of jump in with the graphic user interface, which is really amazing. Those kinds of things will help people kind of come onto the other side of the fence, I think, for sure.
Okay, maybe I have a follow-up for this later, but I'm just gonna ask you, Genevieve, so because you're both helping researchers adopt this pyramid, and you're also coming at it from the other side, or at least you have the ability to talk to people who can make it mandatory as well, right? So,
Yeah.
so where does this impact?
In complement to what Katie said, my role, obviously I'm in the open science program, but I don't play with data myself. I just try and summarize and explain what researchers need to do about it and try to make it easy and accessible for researchers to do so. So I'm more involved in the top 3 steps there. So in collaboration with Isabelle, we have published, for example, an open access publication policy, and we're building a lot of more documents trying to summarize and explain the ethical and legal considerations that researchers have to follow. And this plays in part with the role that I have on the CEO's side, where there are documents that are mandated to support research. Also about the incentives, we're trying to remove as much barriers as we can and try to make it rewarding for researchers and students to engage in open science and data sharing activities. For example, we signed the DORA declaration. We are in the process of modifying the research evaluation, yearly evaluation for researchers, where they can highlight different results that they've published, that research these extra in French, I'm sorry. So basically there used to be only space for researchers to highlight their publications, but now there's a new place for them to highlight other kinds of outputs of their research. And we also have awards for grad students and postdoc fellows that provide financial incentives for the adoption of open science practices. And also finally, the normative community. So I try and be present in communities and events such as today to start discussions between people who may not have had the opportunity before to speak together. And one thing that I've observed, my main pain point around data management and data sharing is that it's more and more required by funders and journals that researchers share their data openly. But the open science and open data can be frightening to some people, especially in mental health, where data is particularly sensitive. And sometimes there's a need for education, that openness is on a spectrum, a continuum. So there's basically totally closed and protected. And at the other end of the spectrum would be public data fully open. But we're working currently in the gray zone and trying to establish what is, the required amount of protection for data, or the minimum amount of anonymization that we can provide to the data in order to still be useful to researchers.
Good. And maybe, yeah, Gabe, I just want to say,
Sure.
So you are almost, I think, to some extent on the other side of this, right? You're looking for regulation, or at least some guidance on what is it that that makes it a working solution, right? So I'm wondering how this impacts you in that way. And
well, I think the challenge that I see with the open science and the open data things thus far is that people end up considering it far too late in a lot of the work that they're doing, because you see, oh, there's a policy that I have to release it or so on. And upstream of that, there were many choices, or that were usually implicitly made in a lot of projects, where there was no organization imposed of any kind. When you go through a project like that at the end, and then you see the data you have, you run into this challenge that all of these things that we'd like to do now require huge amounts of work.
Yeah.
Organizing, choosing, figuring out what you have, what you don't have. So a lot of the things you guys already covered in terms of, Nipoppy and so on, being able to figure out how to do that organization. If you have to do it at the end, then it's huge amounts of work. And so what I've been trying to do, particularly here when I've been involved in new projects of where they're going to collect data, is obviously we're building, open data capture, which imposes structure by dint of it being software in the first place. But in many other projects, just the idea of think about your data capture early, think about your organization early, think about your data model early. And that helps you in a couple of ways. Obviously, it helps from the open, sharing, anonymization, all these things, because you have a model organized and you understand the data just at like a meta level. But then it also allows you to make decisions about what your analysis will be earlier and decide whether or not your analysis is actually sensible earlier. And so that's a big part of that, is that trying to normalize that kind of idea of the project starts. Think about that, semi-technical component of how do you organize the data? How do you store and manage the metadata associated with what you're collecting? Which is a plan. And it's, technically that's the data management plan, which is going to be imposed at some point by the Tri-Council, maybe, but like in the much more in the trenches kind of concept of what do you actually do to do it?
So my first question would be a good segue from what you just said back to Matt, and maybe we start from there. But so what Gabe just said was basically, and we talked a little bit about before this call, that one of the issues is the way we often learn about good data science practices is by making the problem ourselves, right? By making the mistake ourselves. And that is great individually, but bad for science and labs because it takes a lot of time. So one of the things we discussed was like what Gabe was saying, well, it would be nice to get like a big overview of all the different things you will have to deal with even before you learn about them, and then learn why those are important before you figure out by not doing them. So my question is, Matt, you have done a lot of data wrangling, let's say, as part of your PhD. Part of this was adopting a standard, which you've done the bits conversion. And I'm kind of interested in two things. Like one, what was difficult about this? But the other question is, what about this is something that is important for you to learn, or how do you learn it? Because there's always this question, like, is this more like plumbing, or is this more like a microscope, right? Is this infrastructure, or do you need this as your research?
Yeah, so I think the immediate challenge is obviously that like it takes time, and like it's when you're like doing your PhD, you're like trying to meet deadlines or whatever, right? It's hard to convince yourself to like invest the time in something like standardization. Like it just doesn't make sense. It's like I want to get the result and I'm wasting my time like organizing this data. Like why would I do that, right? And I think especially that was more true like in the wild west of bids conversion days, but I think now that there's like stuff like that come to bids and like the stuff that you guys showed, it's like a lot more tangible. But even then, it still takes time to bring your data, right? So then I think I go back to like, why is this worth it? Like, what can you actually do now that the data is organized? And I think that goes back to a lot of what Gabe was saying. So it just like leaves you more open to do stuff down the line where you're not scrambling later on to organize stuff. And I think especially as far as bids goes, it just leaves you more flexible. Like you just have more options of what you can do down the line. So for example, now that my data set's bidsified, it's a lot easier to put it together so we can share it with people. It's a lot easier to run pipelines that are out there, like fMRI prep, for example, that like need bids as its input. You just have like a lot more options with what you can do with it, I find, now that it's organized. So it's like psychologically weird to convince yourself to organize your data when you technically don't have to. It's on my data, right? But I think like once you do, it's like an investment and you can really take a lot out of it. So as it goes on.
Yeah, just to add to that, so you were one of the early adopters, which is we are very happy that you did it. What do you think would take other people in your lab to make the same decision? Like what's the cultural or social aspect of this that will help?
Yeah, it's intercultural.
Is it the value or the easy, basically?
Yeah, I didn't do this because Mulder told you to do it.
You will open your data.
So I'll jump in, and one complication is that a lot of the bids infrastructure is very human centric, and a big chunk now, I'd say probably 80% of the neuroimaging in our lab is no longer human data. It's now animal data. And so that we don't have the same sharing infrastructure for animal infrastructure stuff right now. And, your existing annotation system in Neurobagel is human-centric. And, there's lots of value in that, but we've moved to animal work in a lot of cases where a lot of these features simply do not currently hold any value. And so when that happens, there's the challenge of doing work with no obvious value for doing so in a way that like Matt saw obvious value.
And I will just like quickly add as well that like in our lab, like the people doing the human data, there's like a lot of overlap in the data sets they're working with. So it's like if you have a tool like Nipoppy that just like that can be centralized and track like exactly what person X, Y, and Z all did on that data set, helps reduce redundancy in that.
Yep.
So of the human data there.
Of course, the other challenge with that is that the software we're using actually isn't available in Nipoppy either.
Yeah, I would say a couple things to add to that though. I think like the challenges of sharing animal data are non-existent, right? Like we don't have to worry about consent and like that. We can just we generally post our data sets up on somewhere as soon as they're ready for publication. And that's been one of our practices moving forward. But I think where we would benefit, I think in the animal world, where we are is kind of like 10 years behind like where human neuroimaging is, where we're still developing pipelines for every task that we want. And so one simple one is the rabies pipeline that comes out of our lab, where we're still trying to fine tune what it means to have a good, like what is a resting state network in a mouse, and what does that even mean, and what all the parameters you can fiddle with to get that out, right? And so having Nipoppy actually figure out here are all the things that have been done in this data set. Let's not repeat any of those. Let's just go back and if there's parameters to tweak and things like that, kind of into the development stage of things. And I don't know if it's been conceptualized in that framework, but like across development, but that could be a really helpful utility for us too, kind of as part of a debugging workflow.
Yeah, that's a good way to focus on that as well, is that a lot of the neuroimaging in our lab, and I'm sure in other labs as well, is more experimental in terms of how pipelines are developed and run. And this kind of standardization effort is great when you have a piece of software that is standard, but we're often not at that place yet.
One thing I just want to add on this is I think the concept really is to build communities. So like if the data model that is being used, like in our first kind of use case, like most human-centric and Parkinson things, really the goal is that, okay, there's a community that needs a data model for like a marmoset or other things, and that can be built, and so I think that's and getting still having the flexibility while having the kind of a capacity to share more easily across labs is really the hard kind of balance we get. But I think there's a way out of that problem by community-based data models and
yeah, I think the next question is for Katie. I mean, you're an interesting person because you just transitioned to PI. So I assume there's a change of mindset from going from a trainee to ECR to PI. How do you see the FAIR data and open science as you have now in a different role?
Yeah, so I think what I wanted to mention about some of the discussion that's been happening first is that the normalizing some of these practices is what's needed, I think, to transfer into these better practices, and having people who came up as a trainee with some of this becoming PIs actually is going to help with that. So as a new PI, time, and I said it earlier, but it's really the issue. All of a sudden you're inundated, you think you're busy as a postdoc, but it's not true. And people would tell me that, and I didn't believe them, but it's true. So you have to find ways to integrate these kind of practices, because they can easily fall by the wayside. But growing up, or like career growing up with that as some of your priorities and some of your values, it helps you to not drop them when you have other things that are taking priority. So I think that having that culture in the lab you're in with the PI and with the group and the institution really will help with these things. And also providing training opportunities to students so that they can bring that to their groups as well. And that because the students are the ones, and, maybe some of the staff are the ones that are doing the majority of this work, that they have to believe in doing it and all of that as well. So like you said earlier, it's not Malar that told them to do it, right? So they have to believe it and see the value in that. So I think that the training of like trainees is going to be in the education is important, and having that support from the PIs as well.
I actually have a follow-up question to this because, excuse me, I agree with Nikhil. It's super fascinating to have you as having been not PI and now is PI, because I think the question I always have, and I'm just going to put it very bluntly, is at the end of the day, aspiration doesn't make you change things, I think, right? If you think this is good for science, it gets you a little bit of the way, but your tenure committee at some point, or your grant or whatever, right? Then that's where the real priorities come out. And I think that's valid, and I think it's important to acknowledge this because that's the only way we're really going to make any differences. And so my question is, you said you don't have time, so that sounds to me like make it easy.
Mm-hmm.
The other side of it is, what about it could be rewarding, and not so much in the values that you have, we all have, but really for you, right? What is it that it could fix for you?
Mm-hmm. Well, for one, I mean, there's a lot of initiatives lately that, coming down from the top, that it's required is rewarding in the sense that you have to do this, and it lets you tick the boxes kind of that are required. So that's helpful. But there's also a lot more initiatives to actually reward these kinds of activities, whether it's through monetary awards or recognition or becoming part of a larger group. So I think that that's something that can be really exciting, where open science allows you to kind of work in more closer collaboration with people, and then you're able to maybe work on things that will be more impactful, because when we bring data together that come together through this and other tools, then we're able to actually answer more complex questions in research, and especially in neuroscience, where you're not like one data set that from one whole grant is not actually going to break the barriers of the questions that we're actually asking as PIs. So I think that this kind of collaboration and having data that allows for those collaborations is really necessary to hit the next level in terms of research at a broader level.
Yeah, I was gonna mention that the open science team can serve as well as an organizational memory, because oftentimes each researcher will face the same problems dealing with one process. And what we did over the years is we accompanied several case studies and took diligent notes and made flowcharts and summaries and everything that a researcher will need in the future when they are facing the same processes and they have the same goals. For example, we have accompanied the CRISP Open Data Bank for their REB approval, which was our first open data bank that was approved. And then every other bank that's going to come afterwards will benefit from their challenges and the solutions that we have come up together. And so this will be useful because we have a macro vision of open science and data sharing, and that can help harmonize processes between researchers.
Cool. I'm going to ask one last question to Gabe, and then if you have general questions, we can also go through that. You're in the unique position, almost enviable position for a non-clinical person to be in this thing. What do you think will help? How can we communicate the need for positions like what you have? Because I believe having like a cross lab or institutional level data or informatics centric positions are super useful, but I don't know how to convince people to do that.
Yeah, it's a really tough problem. I think a lot of this originates from if you're a non-technical person, you may not realize there's different kinds of computer people, and they're not all the same, and they have different skill sets. And so, IT is not sysadmin, is not DevOps, is not software developer, is not high performance computing. Those are not the same things. You can have skill sets in more than one, but they're not the same kinds of people. And so it can be hard on the technical, non-technical side, looking at, oh, they just do, you just do computers to do this part of your science, but that's actually could be a number of things overlapping or specific skill sets. So that makes it really hard, I think, for researchers to hire or to find somebody internally who can successfully actually solve your problem. And often what you end up happen is you go and do a hiring call or something and interview some people, and you hire someone and you end up with someone who can't actually solve your problem in the end, even though you hired a quote unquote computer person to do some programming or something. So I think in institution wise, the best thing to do is you need a champion, as is we've mentioned in other cases, who can demonstrate the value of someone like that in general, and then share that person with the rest of the institution with the hopes of growing them out of kind of within lab positioned out. And I say that as essentially that's exactly what's happened to me here. That Millar took me on originally to support his lab, and then managed to grow and build this my experience and my background, and help all comers in terms of whatever technical things needed to be done. And now you have this we're just arriving at the point now where we're saying, okay, maybe we have institutional support here. I want to clarify, we're not quite at institutional support here. We're still supporting this on grants, and the fact that that the people who participate in our grants are being successful, so we can pull more grants, and that's how we continue to exist. But the institution, having the institution step up and support isn't actually quite there yet. So that's the next step still.
If I can just add something that I think that many people inside neuroscience might not be aware of is that outside of neuroscience, the labs don't think they need a computer person. So I think that step has been something that even the neuroscience labs for, a while didn't have full kind of tech, a programmer that's part of the lab. It's just the students that kind of did it going up. So I think that this is something that understanding that there's an importance in all research for a technical support person is really key. And I think this goes really, really broad to the level, of policy and all of this stuff. But it's something that it should become second nature to have, and it's falling on the students, which is great for students to get those skills, but also it does, kind of put the burden on them rather than on the PIs who don't have those skills, because, well, I have a little skills, but for people who didn't grow up with that in their career.
I was thinking, in a way, what you were describing mirrors the question we were asking Matt in the beginning, because this is the long-term pain learning experience for the PI, right? The student has a cycle of maybe 2 to 5 years. PI might have a cycle of several decades, or institute like that. If somebody retires, you learn that it was bad, that was the only person who knows something. But that's very late in the game. Yeah. Do have a curiosity question, but I'm not sure what the time looks like. So maybe this is also
a few more minutes.
Yeah. So the reason this permit is ordered that way and not the other way is that supposedly the way you make change is you go from the bottom to the top, right? And I think often when we talk, and depending on where you're at in what your perspective is, we end up somewhere where some of the bottom exists, but then we say, hey, it's hard, let's do it top down, right? Let's make it regulation. And I think that the problem with that, as far as I can see, is often that you will not make regulation as detailed or as specific as you would need to actually make it useful, because everybody's going to say that's impossible, we're never going to do that. So what are your views on this problem? You said training, right? Incentives, access to community, all these things that are in this permit have been mentioned in a way, but I'm just kind of as an open question for everyone, what do you think we're at maybe, and also what would be the next step that we need to focus on?
I think that we're doing all the steps at once together, so we need to speak more in order to align ourselves together. And also with the CS, because they have all the clinical data that we want to cross with the research data. So we need to have these discussions right now. I see it as a good thing that it's coming from top and bottom at the same time, because it means that everybody recognizes that this is important. But yeah, we need to talk more.
So one other thing I think in terms of what actually drives this, you have to obviously have an availability of infrastructure to people to do anything, but the communities, or the norm, I like to think of it more as like norms as a big key component here. But how do you achieve norms? You achieve norms with champions. You achieve norms with some person in the institution who's doing it the quote unquote right way, the way that we're trying, we're pushing and we're trying to incentivize and so on, who ends up being very successful and who demonstrates to the others within their kind of local area, that when you do it that way, one, you're not punished, and two, you're actually more successful as a result. That has to happen in each of kind of the silos within the institution separately, because there are many silos here within the Douglas. And sometimes they interact with each other, but often they're not really. And some of those silos don't have a champion for this yet, or have just recently hired what we would hope to be a champion. And to have somebody grow up and to demonstrate that if you do it this way, things are better. And so the change happens. And finally, I think the other thing that happens with norms is you eventually have the paradigms of science change with the death of each scientist. And that same thing happens in data management, in methods, in all of these open science things, unfortunately, that sometimes the only way that it's going to happen is the new hires coming in have grown up, as Katie said, within the existing within the new paradigm, or have come in and say, I want to do it a new way. And so they do. And then at the end, you have everyone doing it because they've all been replaced, which is the slowest way that it happens, and you hope it doesn't happen that way, but it can eventually happen that way.
Thank you. I just want to talk about my small experience as a master's degree. I did my previous internships, and I wasn't aware of this problem of data management and public data. And my question was, how can we make it more global? Because in my previous labs, it's not a major topic, and I think it's important to spread this to make things advance and hope to have a good data management and etc.
This is not common knowledge, but there's a whole team at the McGill Library that specializes in research data management, and their services are offered to you for free. So maybe you could start there. And of course, if you have any questions about open science, Isabelle, Katie, and myself will be glad to help you.
To add on to that, I guess this event today and events like this are meant to hopefully change the narrative. Because definitely it's not talked about enough, and also through word of mouth, and also it's going to come top down from the labs. So there's going to be some labs like mine where that's going to be part of day one onboarding, or but then there's going to be others where it won't be as involved. And hopefully with events like the Douglas Research Day, and where all the labs trainees can get together, then it will kind of shift through there, and people will learn through these different events that these are things that are important. And also we have on our website a lot of links to things that are relevant for these kinds of practices. So we try to be active and hopefully get the word out in those ways.
Speaking to the broader question of like, how do you have students in general know about these things? One would hope that students pay attention to kind of the soft skill kind of advertising that happens. Like you get emails from your department, from the university, and so on, saying there's these educational components about things like the data sharing, the data management from the library, or so on. You have to realize that while your core part is your research project that you're doing, there's also this broader thing of educating yourself with the soft skills to go and continue in research. And part of that is learning those things. Those are being offered because those are the hot topics that administratively and institutionally they want people to know about. And so getting yourself in on the ground floor of like what's being advertised and what's being pushed for students to learn about that's outside the lab immediately is a way to see these kinds of things coming. And that's why those are advertised in general, like you're here in general. But in the broader sense for students, it's really, there's other things than just your research. And when you spend some time on those, you can often gain skills that differentiate you as well from other researchers.
So sign up for the Douglas newsletter, sign up for all the other newsletters, and you'll see all this stuff.
So I think we'll have to end there. We've run out of time.
Yeah.
Thank you very much, Nikhil and Sebastian, for coming in giving the presentation.
And thanks for the invite.
No, thank you all for being here.
Thank you for coming.
And since the mental health hospital, I think I'm going to give you all your homework about what they just said. It's like early detection is very important, at-risk people. So if you see at-risk people who are following the wrong path for open science or doing, so find them early, refer to the people who are doing open science so we can have early interventions, hopefully, and a better outcomes in future. So again, thank you, everybody. And yeah, if you have any questions, come find us.
Thank you.